one comment on something I like very much, and one question which I hope might start a bit of discussion. So the comment is that I was really impressed how beautifully this all fits. The second and third Charkas rules, basically a causal explanation for that. The second rule, that means basically that the AT and GC ratios in a single strand are the same like in double strand. And the third rule, that neither theories nor Perigedinská alone. That fits perfectly.
Yep.
And quite an explanation for it. But the question, I have a little bit of a problem with these patterns being necessarily viewed as a code, that means encoding something. Could it be, or why do you think it's a code rather than something like, let's say, carrier wave? Something like a patterning of carriage returns, patterning of pages and numbering of pages in a book, or some kind of infrastructure, let's say.
That would be then a code number 5 in the same sequence. Of course it could be something else as well, but we don't know yet. We didn't see any demonstration of that.
Happy about the pattern, but I'm not sure whether we can talk about code if we don't have the other part, that means the meanings, or arbitrary or assigned meanings.
All right. If you in information theory, when you have a text in which something is encoded, that is, there is a certain rule by which you could read that text and figure out what it says. Then those codes which I listed fits that information theory definition, because they instruct you what these sequences are good for. Right?
Yes.
In that sense, they are codes. I'm not going to semiotics. I'm just very technical, very technical. Here's a sequence. Try to read it, what that sequence tells you. Like if you take, say, a book written in Mongolian, I'm sure that nobody would be able to read that book. And you would think that this is nonsense, this is a garbage.
You don't know it's a book that in Mongolian, it's like
but that book turned out to be a Bible. Simply you don't know how to read that. If you would know the code, then you would know what is written there. If we would know all those codes which are there in the genomes, we would know what is encoded there. All the functions and malfunctions, I don't know, life and death maybe.
It's also a meaning or a function. It doesn't have to encode something specific. It might be something like a parity bit. It also counts.
Well, the codes, since they are degenerate, they could be very different. Like, for example, in a population of flies, of Drosophila, there is a certain amount, like 10 to the minus 5, flies carrying a specific mutation. This specific mutation is interesting by the fact that it is reversible. It may reverse. And the person who discovered it thought that this is just a funny thing, all right, reversible mutation, big deal. But then he discovered that somebody in South America also found that very mutation. It turns out that this small proportion of flies with that mutation is important for population. It encodes some advantage for that population. So the codes could be an immense amount of immense degeneracy.
Actually, I could immediately imagine one reason for one good selective advantage system which for this kind of it's giving the ritual, giving the method to the
All right.
And therefore, we are out of ritual. When I learn something, there's something wrong, virus is there,
exactly.
and we have to be careful.
nature understood the importance of rhythms. There is a 3-base rhythm, there is a 10.4 base rhythm, there is 11 base rhythm in eubacteria, there is a 10 base rhythm in archaebacteria. Nature likes rhythms. Yeah?
So you have shown us the tremendous picture of the multiple codes that are overlapping and so on. And I would like to ask, because the show must go on, and if all these particle codes are already correct, for example, there is also the possibility that we should understand the interactions of these codes that are overlapping.
Right.
And, what is your idea about that?
Right.
What will be the future about it?
Right. Interactions. NF kappa beta factor and many transcription factors, they essentially recognize the same sequence as histone octamers. This is a case of direct interaction of transcription factors with the nucleosomes, with an obvious consequence. They regulate, they stabilize or destabilize the nucleosome. So this is interaction of two different codes. Now, curvature code and chromatin code on the same sequence. If they are changing the DNA axis in opposite directions, then it may mean that while DNA is bound to histone octamer, you better have that DNA bent in opposite direction if you want to detach it. It is very useful when you want to peel it off. But DNA, as soon as you destroy the contact, it goes opposite direction, peels off. This is another case of interaction.
And the second part of the question is that still, I think that if we had these codes that are overlapping, as you showed, it will also it gives some, I would say it gives some structures on the shapes.
Yes.
These shapes are meaningful, let's say, and still, you told that if we have some Mongolian book and that there is no reason that I
Throw it away.
So we do the garbage, of course. On the other hand, we also know that the cell or the photosynthetic transferring apparatus, and so somehow read it, somehow understand it, even that this information is very sparse and so on. So I think that we need something like this, let's say, predisposition or predisposition or some education about some system that will be able to go inside to this and code all these overlapping codes and so on. So what is it? What is this, let's say, subject that is able to do it? Is it cell or is it organism as such, and different meaning of this code?
What orchestrates all those codes?
Yes.
First of all, I think that even for the orchestration, there is a bunch of codes, all written in the sequence. You realize that everything, practically everything, what we know about living organism is there in the genome. Practically. We could speculate that there are some things beyond the genome. But most of it are in the genome. And considering all complexity of living matter, the genome responsible for that complexity obviously has to encode that complexity in that linear form.
Because it was useful to...
Yes, it was selected for. It was selected for.
Maybe it's something older, or somehow adopted from another operating or matter, and using this. The rules are older than the genome, let's say, just a speculation.
Right. Well, I tell you that I think that this tiny amount of codes which I described, it's just even not a tip of the iceberg. It's just one snowflake. One code, I think we discovered just about a few days ago. A very interesting and exciting code. You know about triplet expansion diseases? So triplet expansion diseases are caused by expansion of triplets GCU and also circular permutations of this, which each one in principle encodes for some amino acids. GCC and AAG, Friedrich's ataxia. So they encode altogether 11 different amino acids, taking all those frames. Now, what we discovered is that if you take repeating amino acid sequences, they concentrate in the first exon of human genome and many vertebrate and higher plants genomes. Then we looked at the spectrum of those repeats which are in the first exon. And we discovered that these are those 11 amino acids. Those 11 amino acids. In other words, the first exon is a playground for evolution. The sequences expand and the cell either accepts that or not, playing around with the sequences. When the play is over, they incorporate that piece as another new exon and borrow the sequence from intron. Now I'm speculating. The fact is that in the eukaryotic sequence, in prokaryotic as well, as it turned out, from time to time, here and there, the explosions of little expansion happen. Expanding are only these amino acids. And it started from the beginning of life, according to theory of early evolution. The very first gene was GGC, GGC, GGC in one strand and GCC, GCC, GCC in another strand. It's one of the expansion diseases. So it started with expansion of that thing, then with appearance of membrane or whatever cell, other sequences also got a chance to expand, and they expand since then, selfishly. This is the only meaning of selfish DNA which I accept. These stupid repeats, they are selfish. They explode here and there. Cell either dies from that or selects against them, but they continue, appear and appear, as if life emerging again and again and again in different points of the genome. We call it primary expansion code, because it's a pattern responsible for very specific molecular behavior. Yeah?
By the way, there might be something very special about plants in handling the small repeats. Because, first, quite a lot of plants life depends on polyproline-rich proteins.
Polyproline.
Yes. If you take the polyproline encoding sequences, like in hybrid proline-rich proteins or in formins, usually these are in the protein version. They are something that sticks into the cell wall or resides in the cell wall, actually modifies the properties of the cell wall.
And it's variable.
The genes, that's enormous evolutionary plasticity in the genes encoding this kind of things. And actually, I know that these two gene families, one of them published with focus on this, the other one, so that we came across also, where you have within a polyproline-rich branch of the family, you have a part where you can actually trace down that there is a different repetitive sequence arising by inversion of those repetitive parts, and it's like shuffling a pack of cards.
Yes.
And also those sequences. Yes. Extremely rapid evolution. There's a conserved domain, so that you can still analyze the proteins, and the non-conserved protein, which all sorts of things happen extremely rapidly. Very beautiful system, I think.
Right, very good. Thanks for that. CCG repeating means also CGC repeating. It means also GCC repeating, and this is the most ferocious diseases by GCC in eukaryotes. Well, at least probably they are variable in the copy number.
Yes.
Oh. So this is expansion contraction.
They're also very between even two closely related species. You can't really tell. You can make a good phylogenetic tree on the bonsai plants, but just the heads have almost nothing in common.
Right. So plants are as opportunistic as any living species. And since polyproline happens, plants utilize it. Yeah.
The thing that probably struck me the most was the perfect paleontropicity of the consensus of nucleus constitutivity.
Right.
And which is of double interest because the length of the paleontropy matches one floor DNA, one
Yes.
DNA,
Yes.
universities, it's in base pairs. Do you think, and even more, this is randomly repeated one after each of the nucleus, and I think that there might be some interesting implications in the dynamics of the structure of these sequences, because it's known their encoded information polymorphs are prone to adopt two structures. Basically, the one that is the typical structure, double-stranded, and the other one, the alternative, which is cruciform.
Cruciform, right.
Extruded. And when you have this type, it's randomly repeated. It might, it occurs to me that it's double-stranded DNA.
Yes, yes.
Might not be that rigid as typical random sequence.
Yeah.
Did you think the intentions of this possible application, or did you find something that?
No, I don't think that this cruciform do form, because the nucleosome pattern is very scarce. You never see the whole thing. You see bits and pieces here and there.
Yes. So this is just consensus.
It's a consensus.
Particular motifs are not done.
Right. The symmetry of that, complementary symmetry of that, it's a direct result of complementary symmetry of DNA itself.
Yes.
The diet symmetry of the DNA molecule. The molecule is diet symmetrical, and deformation properties have to be diet symmetrical. Now, one important thing which follows from that, although the pattern is not ideal and not really repeating as such, the important consequence is second rule of Chargaff. You got to have them in equal proportion, complementary ones, and that is second rule of Chargaff. When you observe the complementary proportions in one single strand. We thought about that. We actually were preparing a paper about Chargaff's rule with some reasonable interpretation. But then we bumped into that, and we realized that that is the interpretation.
Actually, there are other interpretations, but they are not in conflict.
No, in conflict.
Interpretations which would actually express a selective value of this even on the level of RNA transcript.
Yes.
Able to take a
RNA hairpins, for example. Yeah, sure.
Some more questions? So maybe we can continue in the pub right now.
Yeah. The pub has so many places.
They are used.
I see. Good.
So thank you for attention and thank you.
Yeah. Not to go into toilet, yeah?
Thank you very much. Hello? A tam někdo? takže bude možná trochu problém.
Kdo má poliploidy?